is a neuron cell body that then is going to continue on to an organ. All right. And so what we're going to do is we're going to use the ganglia as our reference point, and then over here will be our central nervous system. So we're going to have also a neuron whose cell body is somewhere in your central nervous system, and its axon is going to come out and become part of a spinal nerve or a cranial nerve, and then it's actually going to go to this neuron that's in the ganglia. So since the ganglia is our reference point, then we're going to call these pre-ganglionic fibers. And then we're going to call these post-ganglionic fibers. So the basic pattern we're going to look at is using the ganglia as a reference point. And if it's the neuron whose cell body is somewhere in the central nervous system, and it becomes part of a spinal or cranial nerve and goes to this cell body, then it's the preganglionic fiber. Then the fiber that arises in the ganglia and goes over here to an organ is the postganglionic fiber. So preganglionic fibers arise in the central nervous system, and so they are myelinated fibers. So what cells would create the myelin out here in the PNS? Swan cells. And what do we call the junction between swan cells? Nodes of Ranvier. And if you have myelinated fibers with nodes, then what type of conduction? So, somebody said saltatory conduction. So was saltatory or continuous conduction faster? Saltatory. So essentially what we're saying is that we have a faster impulse travel here because we are myelinated. So if you look at postganglionic fibers that arise in these ganglia that are in the PNS, they're unmyelinated seed-type fibers. So if they're unmyelinated fibers, then they would lack nodes of Ranvier, and their impulses would be carried via continuous conduction. So it would be slower over here. So we have a system where two different types of neurons are interacting together, where you get faster impulse travel to the ganglion, slower impulse travel from the ganglion. And then what happens over here is that when we get to the organ, we can either create an EPSP or we can create an IPS. Let's go back to Schwartz. So what was an EPSP? So what does E stand for? Excitatory, post, synaptic, potential. So what does IPSP stand for? Inhibitory postsynaptic. So the E and the I are the critical points of the word. So, for example, if it was your heart, then an EPSP on your heart would make it beat faster. And an IPSP on your heart would make it beat slower. And that's actually how we control respiration, heart rate, and a number of things in our body is by creating these IPSPs or EPSPs through the autonomic nervous system. Okay? So what we're going to do is take this basic pattern and we're going to look at two systems that are antagonistic to each other that have opposite effects. So if the one system has an EPSP on a particular organ, then the other system will create a IPSP on a particular organ. Okay. So what we do is we divide the system into what we call the sympathetic branch and the parasympathetic. And then what we're going to do is we're going to look at this pattern, which we talked about in the basic form, and look at subtle differences between the two patterns. So when we first started talking about the nervous system, we talked about the somatic nervous system. And we said it kind of goes to skin, muscle, bone. And so the pattern, for example, with the skeletal muscle, is that you have a neuron whose cell body is in the ventral gray horn, and then its axon becomes part of a peripheral nerve, and that axon goes all the way to the skeletal muscle. And then all the skeletal muscle fibers that would be attached to each of these synaptic involves would make up a motor unit. So we only have one neuron involved once we exit the central nervous system, go out into the PNS. So what's unique about the autonomic nervous system is what the pattern that we just looked at is we have this autonomic ganglia where we have one neuron cell body. We have a neuron whose cell body is going to be in the gray matter of the central nervous system, and whose axon goes out to the ganglia. Then we have this new neuron in the ganglia who continues to carry the information to cardiac muscle, smooth muscle, would be a couple of examples, all right? So then the ganglia is our reference point, so we have preganglionic fibers, and we have postganglionic fibers. And we said the preganglionic fibers were myelinated, and the postganglionic fibers were unmyelinated. So that's the basic pattern and the difference between the two systems. So we can typically view the sympathetic nervous system as E situations. So exercise, emergency, excitement, and embarrassment. Those are all things that we would see where the sympathetic nervous system has been activated. Okay. And so years ago, and it works fairly well, if you think it through, it was called the fight or flight system. So that would be the emergency aspect of it. So if you were hiking in Glacier National Park in the spring, and you came around the corner in the trail, and here's a female grizzly bear, and she stands up and looks at you, and then you hear something behind you, and she's got a cub on her. You're between the two. Then you would get this enormous response from your sympathetic nervous system. Okay? And it would be fight or flight. And I know what I would do. The fight part would go away real quickly. Yeah. You see, not going to win that one. So anyway, so during sympathetic stimulation, it dominates or overrides the parasympathetic nervous system. So to put that into context, in terms of just one organ, then we'll go back. So the sympathetic effect on the heart is to accelerate heart rate. So believe me, when you saw a bear, your heart would be going. And then what brings your heart rate back to normal is parasympathetic. After the bear goes away. Right? So the parasympathetic nervous system can be remembered by the acronym SLUD, S-L-U-D-D. So S is salivation, L is lacrimation. So what's lacrimation? Formation of tears, yeah. So saliva, lacrimation, urination, digestion, and defecation. So those are the things that the parasympathetic nervous system is involved in. And then there's actually a overwhelming stimulation of the autonomic nervous system that can occur in very, very frightful events. So it's called paradoxical fright because it appears that even though the sympathetic system was activated, you also got the parasympathetic system being activated. That causes you to urinate or defecate when you're so scared. So it's called paradoxical fright. So my mother was an emergency room nurse, and so when I was learning to drive, then she would always tell me, before you get in the car, make sure you avoid it anything you can. Because if you're in a bad wreck, you're going to have paradoxical fright.
I have a very nice relationship.
Anyway, so, and why didn't she know that? Because she took care of people that had been in car wrecks and come to the emergency room. Yeah. So, when you're looking at the sympathetic pattern, then when we're looking at that pattern, then what we're going to see is that in the sympathetic system, these preganglionic fibers are shorter. And in the sympathetic nervous system, the postganglionic fibers are longer. Oops. Okay. So we have short preganglionic fibers. And what they do is they arise in unique areas of the spinal cord. So the sympathetic system is spinal only. And it involves our thoracic nerves. So how many thoracic nerves do we have? 12. So it involves all 12 thoracic nerves. And thoracic nerves follow what? Out. Ribs out to your body. And then it involves the upper lumbar area. So L1 and L2. So we consider it thoracolumbar. So the sympathetic nervous system arises from the spinal cord itself. Right? Then we have these long postganglionic fibers that arise from autonomic ganglia. And we're going to look at a couple of examples, but we have paravertebral ganglia and we have prevertebral ganglia that we're going to look at. And then again, postganglionic fibers are C-type fibers. So non-myelinated fibers. So if we contrast that to the parasympathetic system, then the parasympathetic system has longer preganglionic fibers and shorter postganglionic fibers. So they're mirror images of themselves in regards to how it's constructed. So we have long preganglionic fibers that arise from the central nervous system. And then they arise from cranial nerves. So cranial nerve 3 would be what nerve? Ocular motor, yeah. And so where does the ocular motor go? Where does it innervate? What we talked about yesterday. Your eye. So it goes to your eye. So what is nerve 7? Facial nerve, all right. So what is nerve 9? Pharyngeal. And then nerve 10 is the vagus. Yeah. So with our parasympathetic, then cranial nerves 3, 7, 9, and 10 are involved. And then the other nerves arise down here off the sacral region of the spinal cord and involve S2, 3, and 4. So we say it's craniosacral in distribution. So it's kind of interesting that the parasympathetic system arises either from the brain or the end of the spinal cord, and the sympathetic system arises from the center part of your spinal cord. Okay, so a different pattern to that. And then, as we just said, then we have these longer preganglionic fibers, so we have short postganglionic fibers. Except for your head, there's a little oddball thing going on in your head. So this is kind of a model for what we were just talking about. So remember, when you look at pictures like this, you always remember you're bilaterally symmetric. So if you have something on the right side, you have it on the left side. So what they did is they just pulled the two systems apart, showing them on one side or the other of your central nervous system. But the red and blue would be intertwined on both sides, all right? So which one up here, red or blue, is the sympathetic nervous system based upon where the ganglia are? All right, and so to make sense out of that, the solid lines are myelinated B-type fibers, and the dashed lines are unmyelinated C-type fibers. So remember, the sympathetic system had short preganglionic fibers and long postganglionic fibers. So when you look at how long these dashed lines are in blue, they're very long, so that's the sympathetic nervous system. And the red represents the parasympathetic nervous system. Okay. And as you can see, all of our internal organs are innervated by both systems, where they're going to have antagonistic effects to one another. Right? There is one exception, though, where if you look at the sympathetic side, the blue side, there actually is one organ where there are no dashed lines going to it. And there's only a solid blue line going to it. So that's the one, what's going to be a very important exception to the pattern. So which organ is it? Do you see it? The adrenal gland, yeah. So this little gland sitting on top of the kidney has this solid line all the way to it, and then dashed line internally. So it's the exception to that pattern within the autonomic nervous system. So anytime we have a synapsis, then we have two things that we have to think about. So one is that on the dendrites here, we have to have a receptor. And then from the terminal axon, we have to release a neurotransmitter. So we have to have a neurotransmitter, we have to have a receptor. And then when we get to the organ, the same thing is going to be true. The organ itself is going to have receptors. And the neuron is going to have to release a neurotransmitter, a chemical communicator. Okay? So we have two places where we have to think about, well, what's the neurotransmitter and what's the receptor? So you can elicit a different response, an EPSP versus an IPSP, if you have different neurotransmitters, or if you use the same neurotransmitter but have different receptors. Okay? So the communication key is understanding the neurotransmitter, understanding the receptor. So that's what we're going to talk about now, is kind of what's going on. So preganglionic fibers release acetylcholine. So, and then it's not going to matter whether you're talking about the sympathetic nervous system or the parasympathetic nervous system. The neurotransmitter released by the preganglionic fiber is ACH. So that's a commonality for both systems. Okay? So we release acetylcholine. Right? Now, the receptor on postganglionic fibers is also not going to matter whether you're talking about the sympathetic or the parasympathetic. And we call them nicotinic receptors. And we call them nicotinic receptors because nicotine binds with them. So nicotine is a very potent neurotoxin. So you could, if you took, so with toxins, it's usually milligrams of exposure to kilograms of body weight. Okay? So if you took chewing tobacco and you put it in a jar and you pour boiling water over it and let it set for a day or so, and then strain all the liquid off, you would have a very potent organic insecticide. And then you could spray that on plants that are infected, and it kills the insects quicker. Because it's a neurotoxin. It's a very strong neurotoxin, right? Because it impacts the autonomic nervous system. Okay. So, which is why it's addictive. That is what. So, postganglionic fibers are also going to have a neurotransmitter. So everything's the same over here. So the sympathetic and the parasympathetic release acetylcholine. The sympathetic and the parasympathetic postganglionic fibers have nicotinic receptors. So if we're going to have a difference in the systems, if this is the same over here, then this is where the difference has to be. All right. So with the parasympathetic nervous system over here, then it's ACH. So then the take-home message is the neurotransmitter of the parasympathetic branch of the autonomic nervous system is acetylcholine, right? So that's easy because it's both places. So what we end up with is a different neurotransmitter, and it's actually named from the poison found in poisonous mushrooms, which is called muscarine, and so they're called muscarinic receptors. Right. So muscarinic receptors, and so if you eat a poisoned mushroom, what actually essentially happens is the poison in the mushroom, the muscarine, stimulates your parasympathetic nervous system, puts you in anaphylactic shock that you can die from, because your heart rate drops, your respiratory rate drops, because the effect on respiration and heart of stimulation of the parasympathetic nervous system is create IPSPs. And so it just drops your heart rate out and your respiratory rate. So that's why I buy mushrooms in the grocery store. Instead of go out in the woods and collect mushrooms. Although there are a couple that are like morels. There's only one false morel that's only mildly poisonous. Pardon? So that one's pretty easy. All right? So now we've kind of got our pattern where we have acetylcholine being released here, binding with nicotinic receptor. And then over here we have acetylcholine being released and binding with muscarinic receptors. Now, the thing that's going to be interesting in the future is that we've done a lot more work with the sympathetic branch because it accelerates heart rate, accelerates force of contraction, so then drives blood pressure up. So we've done a lot more work with the sympathetic receptors to understand those because we needed to do that to control blood pressure. So, but we're probably going to find that there's more than one type of muscarinic receptor. Okay. So then if we can contrast that to the sympathetic division, the preganglionic fibers release acetylcholine. So that's what we put on the board, was acetylcholine was the only neurotransmitter between the preganglionic and postganglionic neuron. And nicotinic receptors. So, and again, get their name because nicotine binds readily with it. And so we have these nicotinic receptors for both systems. So then the difference actually arises in interaction between the postganglionic neuron and the organ itself. And so within the sympathetic nervous system over here, then we have NE, which is norepinephrine, that is the neurotransmitter. So we have a different neurotransmitter within the sympathetic system. And we have different receptors too. So we have receptors we call alpha receptors, and we have receptors we call beta receptors. And so we know we actually have two different types of receptors that norepinephrine will bind with, alpha and beta. And that's how we can understand how you can have an EPSP at one organ but an IPSP at another organ. Because you have different receptors that the neurotransmitter binds with. Yeah, so one of the earliest blood pressure medicines, it's still used today if you have trouble managing blood pressure, is a beta blocker. And it blocks beta receptors because if norepinephrine binds with beta receptors, then it accelerates force of contraction of the heart. So we use beta blockers for it. But they have some pretty arduous side effects in some people. So the newer drugs, the newer blood pressure medicines don't use the beta blockers at all because of that. So because we have alpha and we have beta receptors, then we can create IPSPs and HPSPs. And we actually have beta 1 and alpha 1 and alpha 2 receptors. So we actually have several types of alpha receptors. All right. So this is kind of the image that we've created so far. So, and sweat glands are odd. They're in the oddball. So for our purpose, we're just kind of throw sweat glands out the door because they're the oddball. So with the parasympathetic nervous system, which is the bottom, we would have acetylcholine being released by the synaptic involves of the preganglionic fiber and nicotinic receptors on the dendrites of the postganglionic fiber. And then at our organ, we're going to have acetylcholine as our neurotransmitter again, but those muscarinic receptors on the organ itself. And then at the top, we have the sympathetic nervous system where this is the same. Still acetylcholine, nicotinic receptors here. And the difference arises in the organs themselves. So that we no longer have acetylcholine as our neurotransmitter, but we have norepinephrine. And we no longer have muscarinic receptors, but we have what we call adrenergic receptors because adrenaline binds with them, just like norepinephrine. So if you think about that, if the parasympathetic system puts you in anaphylactic shock, then what would be the antidote? And what do people who are highly allergic to things usually carry? EpiPens. So epinephrine and adrenaline are the same thing, just different names. So with the EpiPen is designed to stimulate your sympathetic branch so that your heart rate will elevate again and your respiratory rate will elevate. So that's the whole purpose of that. All right. So the one exception we talked about was the adrenal gland. And what we do with organs is if the organ is, so the adrenal gland is kind of a triangular shaped organ. So if the cells on the inside of the gland look different than the cells on the outside of the gland and have a functional difference, then we call the center of the gland the medulla. And we call the surface of the gland the cortex. So we've seen that in the brain already because we talked about the cerebral cortex as being that surface area of the brain that's highly folded. So when we talk about the adrenal medulla, we're talking about cells toward the center of the organ. Okay. And then they were the exception where we had a preganglionic fiber that went all the way to the adrenal gland. So the first organ that is going to be affected by sympathetic stimulation is going to be your adrenal gland. Okay? And then the reason is because cells in the medulla called chromaffin cells actually produce a neurotransmitter that acts like a hormone in the fact that it's released in the blood and is transferred to neurons via the blood instead of synaptic clefts. And so the compound that the adrenal gland releases is epinephrine or adrenaline. So that's why we have EpiPens, because we can use that chemical to stimulate, to make the body act like you just got your adrenal gland stimulated. And so it initiates a very rapid response from the sympathetic nervous system. And then we have a trace of dopamine as well. So the important neurotransmitter in the brain. And then what happens is where norepinephrine largely targets alpha receptors, epinephrine targets beta receptors. And we actually have identified 3 different types of beta receptors now, beta 1, beta 2, and beta 3, that all have different effects, have different organs. So that's how we can create an EPSP and an IPSP on different organs, is we have different receptors. So what I did is I kind of created a little chart for you. So the way to think about it, and you can memorize this if you want, the best way to do it is to think about what your body would need in a fight or flight response. And if it's something you'd want to accelerate in a fight or flight response, then it's going to be done by the sympathetic nervous system. If it's something that would be an advantage in a fight or flight response, then it would be the parasympathetic nervous system. So my symbols here, this is an EPSP, and this is a IPSP, okay? The positive and negative. Okay. So what we talked about was the parasympathetic nervous system arises from 4 cranial nerves, 3, 7, 9, and 10, and then from the sacral region of the spinal cord. So it's over here, and then the sympathetic nervous system, which is the first column, arises from T1 through L2 in thoracic and lumbar nerves. Okay. So it's not surprising that the radial iris muscle, which is going to dilate or constrict the pupil. Will dilate the pupil. And if you dilate the pupil, are you adjusted for near vision or far vision? So low light, so there's more reflective light the closer an item is to your face. Right? So you're going to dilate your pupils to in a fight or flight response. So you came around the bend in Glacier. Here's that bear. Your pupils are you're going to be looking for that escape route. Okay. And there's no known effect on the sympathetic. There's no known effect for the parasympathetic. And the inverse is true for the circular muscle, which would constrict your pupil. And the parasympathetic causes it to contract, and the sympathetic doesn't. So we have this reverse pattern. And again, just think about it. You're going to dilate your eyes because you're gathering light from distant objects. Okay? So what about the ciliary muscle? What does it do? Changes the shape of your lens and allows you to adjust from far vision or near vision. So in a fight or flight response, your eyes are going to probably adjust for far vision, looking for that escape route. And so remember, in far vision, the muscle is relaxed, so we would want the muscle not to contract, so it would be an IPSP. And then if you convert your eyes to near vision, then it's actually going to be the parasympathetic nervous system, so antagonistic systems, opposite effects. So the good news is if you learn one system, then the other system is the opposite. So that's the other pattern to look at. So cardiac muscle, if you're going to either fight or try to escape, would you want your heart beating faster to deliver oxygen to your muscles, or do you want it to go really slow? It would go faster, believe me. So get an increase in heart rate, increase in force of contraction. So that'd be an EPSP. Your blood pressure would go up, and over here we get a decrease in force of contraction. So IPSP, and your blood pressure would go down. So that's why beta receptors are used on the heart, because they prohibit this from occurring and keep the heart in a state of less forceful contraction, which drops blood pressure. Okay. So which one was which acronym had the D for digestion? The parasympathetic. So not surprisingly, the smooth muscle in the digestive tract is how we move things down our esophagus, churn them in our stomach, move them out of our stomach into our small intestine, move them out of our small intestine into our big intestine, move them through our big intestine. So that whole process is controlled by the parasympathetic nervous system. And so it facilitates that digestive process. Whereas the sympathetic nervous system would tend to inhibit that movement. And the way to think about it is in a fight or flight response, at any point in time you have X amount of energy. And you've got to decide where you're going to use that energy effectively. So if you're getting ready to try to flee, you'd want to move blood from your digestive system toward your skeletal muscles so that you could produce energy for all of your muscles. That makes sense. So you'd actually want to inhibit what's going on in your digestive system.
Kind of like Star Trek.
Yeah.
Would be one that divert energy.
Exactly. Exactly, yeah. So GI sphincters. Sphincters are what close openings. So you have a sphincter at the top of your esophagus, at the bottom of your esophagus, at the exit of your stomach, at the entrance of the small intestine into the large intestine, and then at the other end of the large intestine at your anus. And so if you're not moving anything through your digestive system, then because you don't have smooth muscle contracting, then you don't want to allow things to move out of your stomach, for example, into your small intestine, because they're not being digested properly. So you're going to increase sphincter tone. And so you're going to have an increase in contraction of those sphincters to close them. But if you want to move things in the digestive process, then you'd have to relax your sphincters. So that one's kind of odd in that you get an EPSP, but if you think about why, so you're compartmentalizing things and you're not moving things through the digestive system. Right? Then the gallbladder is an organ that is associated with our liver, and it produces bile, which helps us digest fat. So if you're not moving anything out of your stomach into your small intestine, which is where the bile would enter the digestive system, then you wouldn't want to release bile because you don't have anything in your small intestine for it to work on. So the sympathetic nervous system creates an IPSP so that you don't release bile. And then the parasympathetic nervous system causes contractions of the gallbladder so you can release bile. So again, if you're not doing digestion, you don't want to release bile. All right. Then the pancreas is an organ that sits just posterior to your stomach, and it sits in a little fold of your upper small intestine. And it adds digestive enzymes to the small intestine that helps you digest things. But it also releases hormones that help you control blood sugar. Okay? So there's kind of two things about the digestive system that we could look at. So one would be the release of enzymes, the first thing. So again, if your sphincter from your stomach is tight, so nothing can exit your stomach, then you don't want to put digestive enzymes in your small intestine. Because digestive enzymes digest them. And I don't care whether it's you or the steak you ate, they digest. And so if you have an empty digestive system and you got digestive enzymes in it, then what they're going to work on is the wall of your digestive system instead of stuff inside your digestive system. So you want to close, so you want to keep it tight. So that's why in a clinical environment, if you've got somebody who's been in a car wreck where ribs perforated the smaller large intestine, you get a gunshot wound, a knife wound, where the smaller large intestine has been perforated, what actually happens is the content then goes outside of the digestive system into your abdomen, and all those enzymes begin to digest your organs. So that's why when somebody has a perforated bowel, you have to put a bunch of saline in their abdomen, drain it, a bunch of saline in their abdomen, drain it, and continue to do that so that they won't self-digest. Because if you had a perforated bowel and you had no way of getting medical care you're probably going to live maybe a week, depending on the extent of the perforation, because you'll begin to self-digest internally because of that. So you'd want to inhibit that. And then if you're moving things out of your stomach into your small intestine, then you'd want to release those digestive enzymes. So now the other one are hormones secreted by the pancreas. And so glucagon is a hormone that elevates blood sugar. And insulin is a hormone that decreases blood sugar. So why would you want to promote an elevation of blood sugar in a fight or flight response? Because you want to take the stores of sugar that you had and move them to your skeletal muscle where you can make a lot of ATP to do power strokes, right? So when you're digesting, you're going to be absorbing sugar. And so as you absorb sugar, insulin causes your cells to take up sugar and to utilize it and or store it.
Does the activation of these systems require that there be a connection to the brain? When you remove a chicken's head, it does a lot of these things, but is it just
for a very short period of time?
Is it the nervous system?
You get erratic muscle contraction.
Okay,
but it's not part of the system going. All right.
If you want to read a really gory discussion of that, in France, when they use guillotines to kill people, physicians would be there to grab the heads that have been cut off, and then they would keep track of how long the eyes would blink and things like that.
I'm not interested in the head. I'm interested in the rest of the body.
Okay. So adipose tissue. Why do we have adipose tissue? To store energy, right? So in a fight or flight response, what do we want to do? We want to get some of those compounds that we stored in fat back into the blood so that we can use it to make energy. So it would actually elevate triglycerides in your blood because you begin to mobilize your fat so that you can make energy out of it. So in a sympathetic response. And then the liver, and so there's two words here. So notice at the front of the word glyco and glyco is the same. And in fact, if you look at this word and you take the ESIS off of it, then you have the word glycogen. And glycogen is how we store sugar in a complex molecule. We're not like plants. We don't store it as starch. We store it as glycogen. So if you add lyse to the end of a word, it means to break down. If you add genesis to a word, it means to form. Okay? So during a parasympathetic event where you need a lot of ATP to do a bunch of skeletal muscle contractions, then you're going to break down your glycogen stores. And skeletal muscle and your liver are the two organs that have the greatest capacity to store sugar in the form of glycogen. And so you do glycogenolysis to take it from a complex carbohydrate to simple sugar. But if you're digesting stuff that you're taking in and you're taking up sugar out of your blood because you released insulin, then you would do glycogenesis, which would be take the sugar, convert it to glycogen, so you can store it in skeletal muscle and in your liver. So gluconeogenesis, so the ending genesis tells us we're going to do what? Form or break something? Form. We're going to form something. Neo, the N-E-O is new, and so the word literally translates to form new sugars. And so what we can do is we can use proteins and we can use amino acids through the process of gluconeogenesis to make ATP, and we can use fat molecules through the process of it's a more complicated process that ends up in gluconeogenesis. So that's why if somebody is starving to death, you see some of the pictures out of some of the ravaged areas in Africa, it looks like they have no skeletal muscles on their arms and legs anymore. Because what you'll do is you'll rob all the proteins out of your muscles to supply your brain and your major organs with energy in that process, through this process. So what about your lungs? Would you want, if you're going to be running, you want your lungs to exchange more air or to exchange less air? So exchange more air. So the one thing we can do is we can change the diameter of the tubes in our lungs. So if we make them larger, the air will flow through them more easily. So we would dilate them, airway dilation. And then if we don't need as much airflow, then we'd want to make them smaller, right? So if somebody has COPD and they have trouble breathing, we use inhalation agents that affect the sympathetic nervous system and dilate airways. That's exactly what we're doing with that. All right. In the urinary bladder, where was you? In sludge, right? So when the wall of your bladder contracts, it makes you want to pee. And then you relax your sphincter that you control urine with. Over here, you would want to relax the wall of the bladder and increase sphincter tone, unless you're in paradox with Frank. So what about sweat glands? Now, remember, they were the oddball on that one. So sweat glands, if you're going to produce a bunch of waste energy because you're doing all this conversion of ATP to ADP in your muscle, then what do you want to do? You want to keep your body cool as possible, so you can increase sweat production. And what happens is you release water to your skin surface. When the water evaporates off your skin surface, it carries that heat energy with it. And so that's how it cools you. So if you used an agent that evaporates more quickly, it will actually carry more heat away from your skin at a faster rate. So that's why rubbing alcohol feels really cold when you put it on, is that it evaporates at a much more rapid rate. So it greatly increases heat loss from the body in that area real quickly. So if you actually have, like a child who's got a really high fever, and one of the things you can do is actually put rubbing alcohol on their skin. It will cool their body temperature down. So you can use ice as well. And other things. So the erector pili muscles, what do? They stand your hair up. So why, if you scare a cat, does the hair all go? So it makes it look bigger. And it cushions blows because you got more dead air space between your surface of your skin and the outside of your hair. So believe me, if you came around the bend in Glacier and there was a grizzly bear female, the hair on your back of your neck would stand up. Okay.
That's just something that we're left with.
Yeah. Yeah, that's why we get goosebumps too. It's erector pili muscle. So, what about the penis or clitoris and erection? It's probably not the best way to make yourself look bigger for the bear. So it's actually a parasympathetic response, which is interesting. So if you worked with individuals, particularly males that have had some real traumatic event sexually, then when they get into a situation, it simulates the sympathetic nervous system, and they actually can't get an erection. So they actually are impotent because of that. Okay. So what we have to do to procreate is to get an erection first, and then after we've had an erection, we ejaculate semen, because you wouldn't want it to occur simultaneously. It'd be a great birth control because you wouldn't get it in the vagina as frequently. So it's actually sympathetic stimulation that actually does ejaculation. So once you've gone through parasympathetic, in the early parts of sexual excitement, you convert to sympathetic stimulation. And then that heart rate goes up, respiratory rate goes up. And the same thing's true with women in terms of orgasm is moderated by the sympathetic nervous system. And then what about uterine contractions? So if when a woman is in labor and you look at everything that's going on with her body, is that sympathetic or parasympathetic? Sympathetic, because their respiratory rate will be elevated, their heart rate will be elevated, right? Yeah. Cool. So there's this guy walking in the woods. He comes around the corner, and here's a bear. So the first thing I had to figure out is, was this a grizzly bear or a black bear? Because grizzly bears can't climb trees, but black bears can climb trees. So it's not a good escape from a black bear to go up a tree, because then you go up after you. So if it's a grizzly bear, since it's the difference in their feet and claw structure, then it's a good thing to go up a bear. So this guy comes around the corner, he sees this grizzly bear, she stands up and starts charging him, and he runs over to the tree and climbs as fast as he can. And the bear is at the bottom of the tree, shaking the tree. And he's hanging up there for dear life. So he would be in sympathetic or parasympathetic stimulation. Sympathetic stimulation. So he waits up in the tree for a long time, and then the bear goes away. So he thinks, bear finally went away. So he starts down the tree, and as soon as he gets most of the way down, he sees the bear charging again, because he was just laying over here in the meadow watching him. And so he goes back up in the tree. And then he's up there, and she's shaking the tree, and he's like, ah! So now it's getting toward dark, and he's thinking, can I sleep in this tree? And he hasn't seen the bear for a long time, so he thinks, she probably just gave up on me, so I'm going to come down the tree. So just as he's coming down the tree, he sees the hump on the grizzly bear coming back toward him, and then she's bringing her buddy the beaver. Did it help him out? So all we're going to do in lab tomorrow is the ear. So why do we have these funny things on the side of our head? So it acts like a satellite dish, right? So it collects sound waves, is what it's designed to do. Not everyone is born with an external. There are occasionally babies born without an external ear. In fact, this picture is from a plastic surgeon, and this child was actually born without any external ear. And this is a reconstruction. So we have a hole in our skull through which the air enters, so the sound waves enter. So that hole in our skull is the external auditory meatus. And so your external ear is associated with your external auditory meatus because it's a funnel to funnel sound waves in. So if you went to the MAC downtown and you looked at some of the exhibits that they've had where it was early life in Spokane, they had these long funnels that were curved like this. Those were the first hearing aids. And they would stick the end of the funnel in their ear, and it would make your ear 4 or 5 times larger than it really is. So it would funnel more sound waves into the ear. Yeah. It's pretty cool, right? So what we have to maintain the structure in our ear is cartilage, elastic cartilage, that allows your ear to change shape and spin back. And so that we have cartilage rings. So the outer cartilage ring is called the helix. So if you have earrings here, then they penetrated the cartilage spun in. Those are helical earrings. Right? So we have a second ridge to the inside, which is the antihelix. So here's the helix, here's the antihelix. And then at the top of the antihelix, the cartilage wides, and it creates a triangular depression, so we call that the triangular fossa. So what the antihelix does is create the ridge that is going to create the funnel for the sound. And so the antihelix has a depression on the inside of it that's kind of whirled, so they thought it looked like a snail shell, so a conch shell. So it's called a concha. So that's the depression that the sound waves actually will enter the external auditory meatus with. And then we have a bump of cartilage anterior to our ear, which is the tragus. And then just opposite it, at the bottom of the antihelix, we have a bump of cartilage right here, which is the antitragus. So one of the things we're going to do is work on those structures tomorrow in lab with the ear. And then what we do with the ear is we divide the ear into 3 regions. So the external ear that we just did, and the role of the external ear is to capture sound waves. All right. And so our middle ear is a hollow chamber in the temporal bone, the squamous portion of the temporal bone. And what terminates at the external auditory meatus is a membrane that we call commonly the eardrum. It's called the tympanic membrane technically, right? And what happens, because we have a chamber within this bone, so we have to have a way in which the vibrations created on the tympanic membrane by sound waves can be conveyed across this open space in the bone. So what we have in this open space in the bone are 3 little tiny bones called our auditory ossicles. And so the one that's actually connected to the tympanic membrane is the malleus. The one that is in between is the incus. And then the one that communicates with the inner ear, which we're hearing is called the trochlea, is the stapes. So what we have is we have the outer ear that collects sound waves. We have the tympanic membrane that's set in vibration by those sound waves. Then we have a conduction bones, malleus, incus, and stapes, that transfer the vibration of the tympanic membrane to the cochlea. Then within the cochlea, we're going to use the movement of the stapes, the vibration of the stapes, to create waves in a liquid that's actually inside of our trochlea. And the waves in the liquid are going to cause a membrane to move up and down. And we have little cells that have little cilia on them. And as the little cells get moved up and down by the waves, the cilia bend, and it's bending the cilia that creates a nerve impulse. So the way to think about it is this collects sound waves, this transfers sound waves to our cochlea. And our cochlea is going to convert mechanical waves into nerve impulses. Okay? Yeah.
Is a cochlear implant work? Do they replace the cochlear, or just the amplification?
No, I think I haven't really looked at that, but I don't think they go that deep with cochlear implants, or superficial. So they don't replace it in place, but they replace the function. But I'll look that up. So here's what we were just talking about. Here's our external ear. And remember, we have glands that produce wax called cerumen. And we do that because for most of our history on Earth, we didn't live in nice homes with screens at night. Little critters like to call them holes. And the wax sticks them so that they can't get down your tympanic membrane. Here's the malleus, incus, and stapes. So what you have is you have an airspace here and you have an airspace here. So if you have differences in pressure in these two spaces, your tympanic membrane would either bulge out or bulge in, and it would change the vibration pattern of the tympanic membrane. So we actually have a tube called the auditory tube or eustachian tube that goes from your nose to your middle ear. and it allows you to equalize pressure across your tympanic membrane. So if you get in a plane and you take off and you go up to 35,000 feet, there's less pressure outside the plane. So we're in a pressurized plane, but a lot of people still get their ears ringing because there's pressure differences on either side of the tympanic membrane. The greater the pressure difference, the greater the folding of the membrane or the bulging of the membrane, which is translated to pain. So that's why some people get significant pain in their ears as the tympanic membrane is pushed outward or inward because of the pressure. So this opening in your nose has to open to allow you to equalize pressure. So that's why they tell you to yawn or hold your nose and blow, because it will create a pressure that will open the eustachian tube to equalize your pressure. So with little babies on planes sucking on something, it always drives me crazy because the baby's crying. It's like, give it something to suck on. Yeah. Right? So then what we're going to do is we're going to look at the fact that the stapes fits in an opening in our trochlea. And so it's called the oval window. And so the stapes is actually oval shaped. And what it has to do is perfectly fit in this opening. So there are some babies born where it doesn't fit in the opening and it just vibrates against the outside of the trochlea, and then they will be deaf. They'll have vibrational sound, but not discrete sound. And so what we do is replace the stapes with the titanium stapes, and then they can hear. So if it's a mismatch between the stapes and the oval window. So what we're going to do is we're going to have a fluid that is non-compressible. In other words, you can't take the molecules in the fluid and make them get closer together. So a good example of one you use every day you drive your car is brake fluid. It is a non-compressible fluid. Because if it was compressible, then the amount of power, the amount of pressure you'd put on your paddle would dissipate because the fluid would come closer together. And then your brakes would never come on. That would be a bad thing. So we have a similar fluid in our ear, not brake fluid, a non-compressible fluid. So since it can't compress, it can't absorb the wave energy. So we have to dissipate the wave energy. So just below the oval window, we have an opening, a secondary opening right here called the round window. So when the stapes pushes in the oval window, the round window bulges out. And then when the stapes comes out of the oval window, the round window goes back in. So we use those two windows to create the waves and then dissipate the energy from those. Then we'll pick this up in lab. So bring 6A and 6B.